Kenetoku katoa, greetings. As I said, I'm going to tell you a bit today about flavonoid pigments and try and convey why they've become an important research area worldwide. Here we go. So flavonoids are small plant molecules most familiar as anthocyanins. So anthocyanins are the pigments that provide most of the red and blue colours that you see in your flowers, fruit and vegetables. And they're also the colours you'll see in autumn leaves around you at the moment and when plants are stressed. When we started work on flavonoids in the late 1980s, there were about 250 papers a year on them. And there were a lot of interest from chemists. And also they'd been a good model system for science since the days of Darwin and Mendel. This year we're heading for more than 15,000 publications on flavonoids. And in my 10 minutes, I'm going to try and convey what's changed in that time and why so many people seem to care about them now. So by far the biggest change is that we've come to realize flavonoids are key components of the human diet for health. They seem to provide benefits to a tremendous range of conditions, from diabetes and dementia through to various cancers. It's often labelled as antioxidants, and you'll see them promoted as that, which may have nothing to do with the way they actually have a biological function. And based on all of the publicity around anthocyanins and health, there's been a likewise marketing to eat your colours or to eat more reds and blues, like at lunch today. And then in turn, that's really driven an interest from all the plant breeding companies to get new varieties of fruit and vegetables which have a lot more pigmentation. For example, you will have seen recently Zespri launched Ruby Red kiwi fruit, the new one that's out. And they really think that'll help keep driving their fresh fruit exports, which are going up and up and every year. And also a whole biotechnology field grew up around flavonoids. So just a few examples. In some of our work, we've worked on lots of different technologies for natural food colorants based around anthocyanins. But other colleagues have produced commercial products such as nutraceuticals for enhanced athletic performance, or even natural hair dyes. The classic blue rinse, but lots of other hair dyes as well based on anthocyanins. One of the key contributions we were able to make was identifying the genes which control when and where pigments are produced in plants. So this is work from 2006, I think, quite a while ago now, where we used the classic model system of antirrhinum snapdragon and found what are called MIB genes. So MIB genes seem to control when and where pigments are produced in plants. They're the key determinant of it across everything that you look at. So if anybody did school genetics, you remember doing Punnett squares with big A, little a, that Mendel used, and Mendel's gene turned out to be a MIB for flower colour. So the very first genes identified were MIBs. And now we use markers for MIB genes throughout the world in breeding programs. Within our own institute, we're breeding kiwi fruit, apple, various berry fruit, where we're using MIB markers to help accelerate the breeding program. They're also really powerful biotechnology. So in some early experiments we did, we showed if we took those genes out of snapdragon, put them into petunia, every cell in the plant became pigmented. So you get very high pigment levels. And through our links into some of the US biotech companies we worked with, it generated some interesting headlines over in the States when we were looking at potentially commercializing some of these lines. The gene technology has a really important use that's being developed a lot more nowadays, which is to try and achieve the sort of amounts in the diet of anthocyanins that you need to have a real health benefit and that can be affordable to a lot more people. You really need to get high anthocyanin levels, higher than you get through natural breeding. And that's one application that's being used for a lot. And for example, those genes from Snapdragon, our long-term collaborator, Kathy Martin, made tomatoes which have very high anthocyanin levels. And after many years, she just got permission to start selling them in the States, and they went on sale to home gardeners, selling the seed just last month, and they're selling extremely well so far. We're also able to contribute to some fundamental questions of how plants regulate patterns of gene expression, in particular pigments. So you'll see in nature, in the flowers, tremendous patterning. Cool. And it was noticed that we proposed this could fit some of the Turing and Guihart and Meinhardt models for self-ordering patterns. And we made the proposal that that's how mobile activators and repressors could form these patterns naturally. And that's since been proved experimentally. And with the increase in computing power, people are now doing beautiful mathematical models using MIB genes where they can recapitulate the actual patterns you see in nature just with a model of MIB genes moving from cell to cell. So the final area I'll mention, and the one that I've worked on for the last 10 years now with funding from Marsden, which I've greatly appreciated, is how these pigments help plants cope with environmental stress and how they evolved to do that in the first place. So you'll see around you in nature in autumn, lots of plants going red. And we now know that flavonoids help plants survive lots of different biotic and abiotic stresses. And a lot of our original food crops were actually pigmented and had higher stress tolerance. But we bred those pigments out during the getting our modern cultivated varieties. And from the work that we've done over the last 10 years, we've been able to get strong evidence that the very first flavonoids probably arose around 500 million years ago, and they were colourless, they weren't red pigments, and they were essential for that first movement of plants from the water to the land to be able to withstand UV radiation. And we got this evidence from working on bryophytes, which are the hornworts, liverworts, and mosses, which are the most distant relatives of the flowering plants that people are more familiar with. And I've become a bryophyte convert, a bit of a preacher for, I think these plants are wonderful and underappreciated. They're the second largest group of plants, but I'll just mention two little highlights. They live in the most extreme environments on Earth, like they're the only plants in Antarctic mainland. And sphagnum bogs store more carbon, twice as much carbon as all the forests combined. While studying bryophytes, we found a brand new group of plant pigments called orondins. And we came to the realization that lots of different plant lineages produce different structures of red pigment. And we think they're all red because they share a function in screening white light. So like these are all different species in autumn, all go turning red to screen white light. But I also think a lot of these have evolved to have unique functions in tolerance of environmental stress. And I think if we can understand how these are functioning to enhance plants tolerant to extreme environments, then we can start to understand how to adapt our plants to the extremes of climate change they're going to face. And I'll leave you with this one example. This is a species from the Australian outback that lives around waterholes. And as they dry out, it dries out itself and curls up into intensely pigmented capsules. And people have left them dried out in a bench drawer for 35 years. And then you can add water and they'll grow again. And this is work from my collaborator. He left some dried out for 5 years on a bench, added some water, and in 15 minutes, they've uncurled, they're still green, and they're growing again. And the key thing there is probably those intensely pigmented cell wall pigments that we discovered that are screening out the white light, but also giving defense against pathogens and maybe water loss as well. So I'll end with thanking all of my colleagues who've made the work possible and fun over the many years, in particular Kathy Martin, my mate across the ditch, John Bowman, Nick Albert, who will be known to a lot of you, and also my most frequent co-author, Kathy Schwinn, who also happens to be my wife and in the audience today. So thank you. the great majority of them aren't coloured.
I was just wondering, do you know much about, especially in that new class, and fluorescence or electrochemistry? Some of those structures you can see.
Yeah, people are interested in that. One of the key differences about those set of pigments we discovered is they are fluorescent. So people are interested in using them as fluorescent tags, for example, in cell chemistry. And also there's people synthesizing them and looking at them as colorants in various applications, or how their photochemistry is quite different to anthocyanins. They've been studying how the charge moves, because they do change color with pH and they change fluorescence with pH, but on a quite different process than anthocyanins do. Anthocyanins change colour with pH as well, as you might know if you've got red wine and put a bit of acid in it or a bit of alkali, it changes colour. But no, it's really interesting chemistry from them, yeah. It's hard to get them in large amounts because they're intensely, strongly bound in the cell wall. And so we think that's key to how they provide extra benefits for liverworts that anthocyanins don't.